All right, hello everyone, and welcome to the Cali PA for another research seminar. Before we get started today, I'd like to make a safety announcement. In case of an emergency, please exit out the hallways and down the stairs. They'll be lit up with exit signs, and please proceed out the building and towards Cesar Chavez Park, the evacuation point. And that is all. We'll get started today. Here's Lori.
All right, thank you, Reed. So I'd like to welcome everyone to today's research seminar. It's titled The Effects of Ultrafine Particulate Matter Exposure in an Animal Model of Neurodegenerative Disease. The principal investigator for this study is Dr. Arthur K. Cho, and he is at the Fielding School of Public Health at the University of California, Los Angeles. His co-investigators for this project are Dr. Michael Kleinman from the University of California, Irvine, as well as Dr. William Meliga, who is also at UCLA. My name is Lori Miyasato, and I'm with the Research Division at the Air Resources Board, and I am the contract manager for this project. Next slide, please. Next slide. Thank you. So another little set of announcements. The speakers will be answering questions after the presentation. So for our online audience, if you have any questions, please email them to me at the email address shown in the first bullet, lori.miyasato@arb.ca.gov. If you'd like additional information about today's speaker or a link to today's slides or the final report, you can find them at the URL listed under the second bullet that ends with cho.htm. For more information about upcoming or past seminars, as well as for a link to sign up for the distribution list for seminars, the link is shown under the third bullet that ends seminars.htm. And finally, if you're interested in learning a little bit more about air pollution's effects on the brain, CARB has developed a fact sheet, and it is available at the web link that you see listed under the fourth bullet. Okay, next slide. There's a large body of evidence that has shown that air pollution exposures are associated with adverse cardiovascular and respiratory health effects. However, less is known about air pollution's effects on the brain. The U.S. Environmental Protection Agency and the Health Effects Institute both did exhaustive literature reviews on the effects of particulate matter exposures on health, and both noted that there's a scarcity of research looking into brain effects, so they both recommended that more research was needed in this area. So CARB's rationale behind this study is that we're interested in determining whether ultrafine particulate matter exposure accelerates or worsens the symptoms of Parkinson's disease. And you'll be able to hear more about how that question was answered in today's seminar. And so finally, I'd like to introduce today's speaker, Dr. Arthur K. Cho. He is an emeritus professor of molecular and medical pharmacology and environmental health sciences at the UCLA Center for Health Sciences. His research has been focused on the relationship between the chemical properties and the biological effects of compounds that are related to the amphetamines and the quinones. His studies have led to the development of assays for reactive chemical species in collected air samples. So now, if you could please help me welcome Dr. Arthur Cho.
Thank you, Lori. By way of introduction, and this is the slide that introduces our group, who includes myself, Bill Meliga from UCLA, and Mike Kleinman at Irvine. The project that I'm about to describe, or we are about to describe, I should say, was designed to test the hypothesis that the reactive components in ultrafine particles of ambient air could induce the development of Parkinson's disease-like behavioral and brain pathology in a strain of transgenic mice that express human alpha-synuclein, which is a protein marker for the disease. So in the study then, both the transgenic and wild-type animals were exposed to concentrated ultrafine particles generated by the bases, which is an instrument designed by Kostas Siotos at USC, which concentrates ambient particles and allows their introduction into, in this case, cages to which the animals are exposed. And the animals were then exposed for 22 weeks and their behavior and neurochemical alterations examined following the exposure period. So the way we organized the project is by 4 different tasks. The first task was the exposure part, which in which the animals are exposed to transgenic and wild-type animals exposed to the concentrated ultrafine particles for 5 months. And this task was led by Mike Kleinman. In addition, then, in the next task, we determined the motor and cognitive behavioral behaviors in the exposed animals before and after, I should say, the exposure period. And Bill Meliga was the person responsible for that task. And then in these animals following their exposure, their brains were examined for neuroinflammatory and neurochemical defects, and both Bill and I were involved in that study. Finally, we characterized the exposure area in terms of the properties of the ambient PM2.5 particles, which were concentrated by the bases, and our laboratory was responsible for that operation. So I'm going to turn the mic over to Mike Kleinman, and he's going to talk to us about exposure. Mike.
Good morning. If we move to the next slide. This is a diagram of the concentrator system, and just to briefly show you how it operates. Air is drawn in into the system. It gets hydrated with water vapor. And this causes the ultrafine particles in the air to accrete water, and they get to grow. The particles are drawn up through a chiller, which is operating at a very low temperature, and this causes the water vapor to condense on the particles, and they go to a size that actually have enough inertia that they can be pulled through a virtual impactor. The way a virtual impactor works is we are sucking air through the system at about 110 liters per minute. And that accelerates the aerosol mass and aims it at a small pickup tube. That pickup tube is drawing air at 10 liters a minute. And so the particles are actually concentrated out of a total volume of 110 liters a minute down to about 10 liters a minute flow rate, which is approximately a factor of 10 increase in particle concentration. The particles are then separated out, and we have some going to monitors so that we can measure particle counts, particle mass, and we used also an aerosol mass spectrometer to do some chemical speciation as a function of particle size. The remainder of the air is drawn through selective impactors so that only the ultrafine components are able to go through and get to the chambers with the mice. At the same time, we also have mice exposed to purified air. And so we collect particles during the exposure on quartz and Teflon filters, and these are analyzed for particle mass, elemental carbon, organic carbon, and trace metals. Next slide, please. This gives you an idea of the exposures. Now, we actually had 4 separate cohorts because it was necessary to break the groups up so that we would be able to actually do the biological assays in a reasonable way. We could not handle all the animals all at the same time. We spread this out over 4 cohorts. And I want to point out that we're showing mass concentration on a log scale. So although the ambient air looks like it's half of the ultrafine concentrated particles, it's actually a factor of almost 10 lower, which was our design. So when we looked at the filtered air for the controls, you can see that there were relatively few micrograms per cubic meter in the controls. And we were operating at about 80 to 90 micrograms per cubic meter. And each of the 4 cohorts were exposed basically to the same concentration. There was a little bit, as you can see, of week-to-week variation, but not very much. So over the course of the 22 weeks of exposure from each cohort, we really got approximately the same level of exposure. Can I have the next slide, please? We analyzed the particles for, and okay. We also looked at elemental carbon, EC, and organic carbon, OC. And you can see in the air group, we have relatively small concentrations of these materials. They should be in micrograms per cubic meter. And the concentrated ambient particles, which were ultrafine, had approximately 15 micrograms per cubic meter of organic carbon and approximately 8 micrograms per cubic meter of elemental carbon. The elemental carbon is essentially graphitic type carbon, and the organic carbon represents organic chemicals that had coated the particles. The trace metal concentrations were measured using an ion-coupled plasma mass spectrometry system, very sensitive. And as you can see, the trace metal concentrations were reasonably low, but keeping in mind that this is particles concentrated by a factor of 10. In ambient air, these would be a factor of 10 lower if you were trying to draw a comparison. But you can see that the major elements, sodium, which is produced mostly from sea salt spray from the oceans. Aluminum is a very common metallic component of crustal materials, and certainly iron, which is a product of both man-made materials and natural products. Those were, the elements that were present at fairly high concentration. Can I have the next slide, please? So these charts are showing concentrations of ultrafine particle mass versus concentrations of specific chemicals. So we're looking at carbonyl compounds, which are aldehydes and ketones, and we're looking at alcohols in panel B. And we're looking at different size fractions. So we're looking at the ultrafine particle mass, which is running at, these are the particles smaller, in our hands, smaller than about 106 nanometers in diameter, as compared to true ultrafine. People draw the line at 100, but the inertial separating system and our capture system really had a median of about 165 nanometers. But what you can see is that there is a relationship between the particle mass or the ultrafine particles and the amount of carbonyls and also the amount of alcohols. Now, these are organic compounds, which are part of that organic carbon fraction. The lower panels, C and D, represent what we would have seen in the particles larger than 165 that are up in the accumulation mode. So these represent particles bigger than 100 nanometers, but smaller than about 2.5 microns. And you can see that there's also a relationship with the mass. As you go down to particle mass concentrations that are lower, that relationship is less preserved. Next slide, please. This is just a point of interest that when we look at the ambient temperature, because we have day-to-day variations in temperature over the course of exposure. And it turned out that when we plotted ambient temperature versus particle mass versus carbonyl concentration and versus alcohol concentrations, on days with higher temperatures, higher ambient temperatures, we had higher concentrations of particles in general and reactive organics in particular. Now, I want to point out, it's important to keep in mind that the exposures all happen at constant temperature. This represents the ambient temperature. So these are the temperatures at which the particles were created, and not the temperature that the animals were exposed to particles in our laboratory situation. And so I believe the next slide is back to, oh yeah, let me just thank, a lot of that data was accumulated and analyzed by Lisa Wayne, part of their UCI, and using the Irvine mass spectrometer, which made it possible for us to really get some chemical speciation, both as a function of day-to-day temperature and size. And okay, back to you, Art.
Thanks, Mike. If there are any questions, if you could hold them till the end of the talk, as Laurie indicated, and then we can answer them all at once. Now I'm going to switch over to some slides over to Bill, who's going to carry on.
Okay. All right, so the main focus of the study then was to expose animals to this ambient air that's been concentrated by the bases and to explore behavioral and brain pathology. And so our initial hypothesis was that combination of factors that Mike just elucidated would, upon exposure to the mice, result in brain inflammation, changes in neurochemical content, and accumulation of human alpha-synuclein in the transgenic mice that we were studying here. So in respect to that, the animal model consisted of a strain of transgenic mice on a C7 background that were expressing the human alpha-synuclein, which, as we know, is considered a primary protein marker that accumulates in Parkinson's disease. And there's a huge amount of research going on in terms of what form of alpha-synuclein is the culprit that's likely contributing to neurodegeneration. There are oligomers and particulates of forms of the alpha-synuclein in addition to a cytosolic form, all of which are still not fully defined in terms of their functions. So with that, we then had to decide how to design the study, which posed a significant challenge in itself because we needed to address exposures or exposed animals versus ambient air animals, and likewise how to accommodate large sample sizes. So with that, we relied on prior data in the literature that, I'm sorry, I'm getting a little ahead of myself. We'll get back into this where we did both motor tests and brain assessments. I'll be enumerating each of these in much more detail. So getting back to my point as to how we, what guided our selection of the time frame for the animal's exposure, we relied on a prior article that had showed in this strain of transgenic mice that in their brains, be it hippocampus and striatum with higher levels in the hippocampus, that there was an age-dependent increase in alpha-synuclein over 3 to 12 months. And so our challenge was, where do we come in with our study in terms of the duration of the air pollution exposure? And other options were to come in before the accumulation of alpha-synuclein was apparent in these animals, with the hope of then accelerating its first appearance, so to speak. Another option was to come in after the alpha-synuclein was fairly well expressed, as in the older animals. The concern there was that with it already being at markedly high levels, we might not see a further increase in alpha-synuclein. So we opted for the mid-course, coming in when the animals are just starting to have this or show this accumulation of alpha-synuclein. And so starting the study period around 4 months, continuing to 9 months, the hypothesis was that we might accelerate this market increase to that's observed at 12 months. So that formed the basis for our starting the animals at an age of about 16 weeks to enter into the study. This just to show how we accommodated some 90 animals in the study. As Michael mentioned, we can't study them all at once, so we needed to break them down into cohorts. And we had 4 cohorts. Details on this is all hidden behind our images here. But essentially, each cohort consisted of 4 groups. That is, we had alpha-syn and wild-type animals, and within each of those groups, we had exposure and air as subgroups. So we were able to have animals enter into the study in these cohorts, all at 16 to 17 weeks of age, and then they proceeded through 22 weeks of exposure, as you can see here in the columns. And during that time, it was for an exposure of 5 hours a day for 4 days per week. That was consistently maintained throughout the 22 weeks. And we had some minor changes here for the holidays, but we still maintained that exposure paradigm. And at the end of the 22 weeks of exposure, then the animals sequentially went through a week of behavioral testing, immediately followed by sacrifice for brain dissections. So this was just to show you then, again, that how we allocated the animals across the 4 cohorts, and that for the most part, all animals are getting exposed to the same time frame, with the early part of cohort 1 and the latter part of cohort 4 being outside of that. But for the most part, we were able then to accommodate, like I said, 92 animals essentially receiving the same exposure conditions for the same time of year. And the other factor was that when it came time to sacrifice, obviously we couldn't have all the animals being sacrificed at one time either. So this allowed us to have some distribution of the animals across these 4 time period slots. But all the relative time frames were consistent across all 4 cohorts. In that week prior to sacrifice, the animals were subjected to behavioral testing as well as prior to the study, but the key tests were post-exposure, and they consisted of both motor and cognitive tests. And these were, the tests selected were based on a huge amount of prior literature suggesting that, or showing that dopaminergic deficits resulted in decreases in performance across these different motor tests, as well as the cognitive tests. And again, I'll go over these in more detail, but essentially they all had a common theme of assessing basal ganglia function, which means the nigrostriatal dopamine pathway, and also fine motor coordination, as well as a species-specific tests in terms of building nests and the like. So now starting off with the behavioral responses that we had with the pole test, where the animals are placed on top of a pole, and the time to rotate and the time to descend the pole were the outcome measures. And you can see then across all our different histograms here, there were no differences, be it in rotation or in descent across the 4 groups, as wild type, air and exposed, and alpha-Syn, air and exposed. So not much to comment on that other than the fact that we could not see any significant differences. And in retrospect now, we're finding, after we had characterized the brains, that this test is sensitive to dopaminergic deficits, but as I'll be showing, we did not measure striatal dopamine decreases, so that's the likely explanation for the absence of changes in this test. The next test was call it challenging beam traversal, where the animals start at one end of this beam that's on an upgrade, and they have to proceed willingly to their home cage, as it were. And we measure then the errors or the foot slips, both in the front front limbs and back limbs, as well as the time to traverse from beginning to the end of the beam. And here we analyzed the data both for front and rear, and air and alpha-Syn and wild type. And as you can see, the results for the air and wild type in terms of the front paw slips were no different across time. So we did it before the study and after the study, and both alpha-Syn and air exposed animals behaved similarly in terms of their front paw placements on the beam. Where we did, though, detect differences in these same animals, then were in their hind limb or rear paw performance, if you will, in terms of foot slips in the hind limbs. And in those cases, then we saw a significant increase in the final versus initial for the alpha-Syn animals, as well as for the wild type, that wasn't statistically apparent in the front paws for those same animals. So, again, the issue being here, we seem to have differences that could be attributed to the the air pollution exposure, but it was not a function of the alpha-synuclein. And in this uppercase and lowercase here, these are where paired analysis, where each animal is evaluated before and after treatment. And whether you take that analysis or just the group means of that same data here for the back final, we detected a significant difference of about 30% more deficits or motor slips in that task. So this was, without going into what brain correlates may or may not have been present, others have commented that this challenging beam test is a very sensitive test for detecting early motor deficits. And it appears that it evolved out of this air exposure, the air pollution exposure, and not in the air exposures itself. So as to what its cause was, we were not able to attribute any type of mechanism other than this observation. Looking for other observations that we went to, this more of a species-specific task where the animals will tend to take nesting material and build a nest over time. And we try to grade that both at an early time point of 2 hours and a late time point of 18 hours to see if they were interested in building a nest and then how well they built the nest given sufficient time to do so. And again, across all four subgroups, we saw no differences in terms of the relative improvement in their nest building between 2 and 18 hours. So all four groups essentially showed a better nest at the latter time point. And again, the statistics did not show any differences between these. So again, in retrospect then, when we, as we'll be showing with our brain analyses, this test too seemed to be dependent on a dopamine a deficit or a decrease in dopamine content in the striatum, which we did not detect. And that again, we attribute then to, we use that as an explanation for perhaps why we did not see differences in this behavioral response. And lastly, we went to a cognitive test where we used a standard Y maze. And here the challenge was for the animals to explore consecutively different arms of the maze in sequence, and that would be recorded as an alternation and be graded relative to the total number of arm visits. And so that that fraction of sequential visits, that is not visiting the same arm twice, but as illustrated here, ABC would be an alternation, or BAC would be an alternation in terms of the sequential exploration of each arm. When we looked at that as a function of total arm visits, then again, we saw no differences across the four groups, as you can visually see by the histograms. Nonetheless, the overall performance of this in terms of the percentage of alternations was matching prior data in the literature. So again, we just have to attribute the absence of any type of inability to explore new environments as being reflected by the absence of brain changes. And as a consequence, didn't see any changes here in this test. So we're using sort of the same argument both ways. For most of our behavioral tests, we did not see changes that we attribute to the absence of brain deficits. And conversely, asserting that the change or decrease we saw in the challenging beam was in the absence of a brain change, and or in terms of these more static markers that we'll be enumerating. But it may be related to some functional deficit, which wasn't part of our analysis scheme. So moving on then to the brain responses that we were going to, that we had hypothesized may correlate with the extent of behavioral deficits. We had a range of inflammatory and anti-inflammatory markers, TNF-alpha, IL-6, HL1. In the alpha-Syn animals, we track the human alpha-synuclein, which was being expressed uniquely in those animals. I should say parenthetically that for that measurement, we verified both by PCR in the initial, obviously the initial organization of the study to have the 4 groups, to have the alpha-synuclein animals segregated from the wild types, but also by the ELISA assay at the end of the study, all our animals that were in the alpha-synuclein cohort were positive for alpha-synuclein. And as a cross-check for that, the wild-type animals were all negative for human alpha-synuclein. We did not monitor the mouse alpha-synuclein. And out of the 92 animals, there were I think 4 animals that were mislabeled, but through that correction scheme of doing their alpha-synuclein with their ELISA assay, we were able to put those few animals into their proper categories. The HPLC method is standard protocols for measuring dopamine and norepinephrine in those brain regions. So then the question becomes, why did we choose those markers in particular? We'll pick up on that.
So what this slide is trying to summarize the movement of the particles and their associated, the particles and their reactive chemicals as they enter the lungs, and then from the lungs they enter the circulation, and they cross the blood-brain barrier. Or some of the compounds can cross the blood-brain barrier, others can't because of that barrier. The blood-brain barrier is basically a membrane system, and it allows the transport of specific substrates for available transporters and the passive diffusion of typically organic compounds that are nonpolar. So polar high molecular weight substances are typically excluded by the lack of permeability, so to speak, of the blood-brain barrier, and I want to comment on its permeability changes as a result of some of the reactive species that are being introduced by entry of the particles into the lungs, where they interact with components of the lung lining fluid to generate reactive oxygen species, as well as inherently reactive electrophiles that are present in the mixture of the particles. And once they enter the lungs, of course, they interact with cells that are in the lung lining fluid or in the periphery of, or in the membranes of the lungs, and they can initiate then reactive species or initiate responses where because of associated stress, inflammatory cytokines, or for that matter, adaptive proteins can be expressed and modify then the overall effects of the reactive species that are present, and add to the reactive species that are present by an indirect effect through the released cytokines. These same cytokines would be present obviously in the circulation, as with these reactive oxygen species that are generated by pro-oxidants and electrophiles. One of the things that some reactive oxygens, or a hydroxyl radical to be precise, does is cause lipid peroxidation. That in turn modifies lipid membranes and alters permeability. And there's evidence also that an inflammatory response at the blood-brain barrier also increases permeability. So the net effect of the appropriate concentrations of reactive species and cytokines would be to increase brain permeability and allow greater penetration then of those substances that are normally excluded. And the consequence of that would be then to enhance the stress-related inflammatory response through oxidative electrophilic stress. And the resulting promotion of an inflammatory response would result in an increase in two inflammatory markers, TNF-alpha and interleukin-6, which was two of the markers that we assay in the brain.
Okay, Bill. So in terms of the brain dissection techniques, that was we perfused the animals to remove the blood component, and then dissected the brains and removed the hippocampus and striatum. Those were our regional reasons for analysis based on those areas or those regions having the higher or highest alpha-synuclein content, with the hippocampus having a higher content than the striatum. But to do so, we needed to work out some assay conditions, and with the TNF-alpha, it appeared that with our assay kit conditions that we needed to dilute our homogenate to get reproducible readouts of the ELISA assay. And it's shown here on the left that we had to dilute our homogenate fourfold in order to begin to get a good linearity in terms of further dilutions in terms of recovery. And this became, it's also shown here that once we diluted in another experiment, our conditions were fourfold diluted, we could then recover any spiked amount, essentially about 100% recovery. However, with that dilution, it took us below the sensitivity of the assay, or at borderline sensitivity. So the bottom line being we were unable to get reproducible TNF-alpha measurements from our brain samples. And the sort of same image or same result appear here with the interleukin-6, though we do seem reluctant to use the word trends, but I'm going to almost in a semi-meta-analysis notion that it shows up across most of our readouts that we do have apparent increases with the exposure conditions versus the air conditions, but none of them were significant. So again, here in both exposures, wild type and alpha-Syn, they're higher than the corresponding air conditions for a hippocampal IL-6. We thought we could try to pool the hippocampi from multiple animals to try to increase our readout values, but that essentially showed the same result, which I believe is not showing up on our screens now. So the next one, heme-oxygenase, was similar. I don't know, Art, you want to explain a little bit more on the details of what you saw here?
Only that there didn't seem to be a statistical difference. Although, if anything, one might expect the heme-oxygenase levels to be a little lower than those animals that were expressing an inflammatory response, because there seems to be an inverse relationship between the levels of HO1 and TNF-alpha. In other words, between the balance between a pro and anti-inflammatory response. And so in this case, one might have expected a decrease in HO1 in animals who were demonstrating an inflammatory response.
And that would account for the exposure conditions here being slightly lower.
Slightly lower, yeah.
We're not significantly different. I should say also that part of the challenge here was with all these multiple assays and the small size of the mouse brain regions, we had to rely on both bilateral dissections, but still distributing that material in such a way that they could be used for all these assays. And that was then apparent for striatal dopamine or dopamine content in the striatum and norepinephrine content in the hippocampus. Again, here our mouse values were comparable to those in the literature, but showing no significant change. So again, you might try to see a small decrease here, but again, that trend was not significantly different for dopamine content in the striatum. So unfortunately, not much to comment based on that those readouts. In terms of the alpha-synuclein animals, then we looked at their content in both hippocampus and striatum, the relative values corresponding again to what was in the literature in terms of the expression being much higher in hippocampus, and that was proof. We established that as well in our animals. And again, we had a slight increase, but was not significantly different in terms of readouts for that. And striatum, we couldn't see even a trend of a difference. So again, I guess not much to comment on because of that finding, other than, like I said, the alpha-synuclein content was what was expected in terms of literature values. So all we can conclude from that is that the exposure conditions did not result in significant behavioral and brain pathology, and there are further issues related to that. We intentionally went for a relatively long exposure period, that is 22 weeks, which was a challenge in itself just to maintain, just logistically speaking. But that in itself may still not have been long enough to induce a frank behavioral or brain pathology. And also during that time with this, what we are be showing in terms of the chemical and biological characterization of these particles, the animals through this exposure conditions may have actually become tolerant to the exposure and thus not showing effects due to that buildup of tolerance and sort of compensatory mechanisms, both functional and biochemical. That's just going out a leg in terms of speculation, but if we had our druthers, we would want to continue that exposure condition for another several months to really ascertain whether a longer-term exposure would result in behavioral and brain pathology. And conversely, in looking at the literature where there's a more toxic AP exposure, conditions that do produce those behavioral and brain pathology deficits, I think that one has to question what is the relevance of those to the human, because obviously we walk outside every day and within a couple months of exposure, we don't seem to manifest frank behavioral or brain changes. That is, if to whatever extent this does occur, it happens over a long period of time. And even with the relative 2-year time frame of a mouse lifetime, we still are coming in with maybe something like reaching adolescence in the animal's total lifespan in terms of exposure conditions, exposure duration. So so while we did not expect or even desire to have large differences, we were hoping to sort of come in between those, come in between such large differences and no differences and start to show minor differences in both behavioral and brain as the beginning of, or the evolution of pathology due to this air exposure. And for one of our tests, I believe we did see that, and whether that's suggestive of early onset motor disorder would be something we would leave as a hypothetical. But more so, I think it's pointing towards the direction that future studies of this nature should be focused more on functional outcomes rather than frank or direct measures, which really relate more to a toxic exposure environment. We've seen this with drugs of abuse as well as MPTP, other models of Parkinson's disease, wherein you do get dopaminergic deficits, increase and decreases in behavioral performance, but only with significant dopamine deficits manifested in the brain. And without that, then it correlates then with our absence, for the large part, in large part, the absence of behavioral deficits. So with that, then the question becomes, well, what did we expose the animals to in terms of the, or can we interpret better the absence of changes in our animal model based on the chemical and chemical properties of the particles themselves.
So I'm going to finish up with the discussion of then what we mean by reactive chemical species, because basically our hypothesis is that there are reactive chemical species in this in the ultrafine particles that are being introduced into the animals, and our notion is that it's these reactive species that are initiating the responses that we're seeing. So by then starting here at this end of this table and going to the other end, I'm going to work my way across with the two categories of compounds. The first category are pro-oxidants, and by pro-oxidants we mean those substances which are capable of performing an electron transfer reaction between the electron source and oxygen to generate two reactive oxygen species, hydroxyl radical and hydrogen peroxide. And hydroxyl radical is a highly reactive oxygen species, so to speak, that reacts with available sites at diffusion rates. So an example of one of its targets is the double bond system of a fatty acid, in which this allylic position is particularly susceptible to peroxidation. And this is the reaction of lipid peroxidation, which is a very common reaction when fatty acids are exposed to hydroxyl radical fatty acids such as those found in membranes. And the result of this reaction is that this fatty acid is cleaved and otherwise modified such that then the resulting membrane can be altered and this membrane permeability can be altered to increase the permeability of a particular cell. The second class of reactive oxygen species is hydrogen peroxide, which is in fact also a cell signaling molecule, but its characteristic is that it will react with protein nucleophilic functions such as a protein thiolate to form the corresponding sulfenic acid. And this protein modification, which in fact is reversible, but once modified, the resulting protein, for example, in a protein such as TEAP1, which is a regulatory factor for the NRF2 antioxidant response element, will activate a site on DNA to increase the expression of adaptive proteins such as glutathione transferase and glutathione synthase, and as well as heme oxygenase. And we've used heme oxygenase as a marker protein for its activation. Alternatively, this thiolate could be that which is on I-kappa B, which is a component of the NF-kappa B system, and activation of the I-kappa B protein results in the activation of an inflammatory response. So this is a pro-inflammatory system, and its marker is TNF-alpha. The second class of compound that we're concerned about are electrophiles, and they form covalent bonds with tissue nucleophiles, in this case, again, a thiolate to form an adduct. Now, the difference between this adduct and this adduct, so to speak, is that this, once formed, is not reversible. So this means that the protein that is modified by the electrophile is irreversibly modified, and for recovery of this protein, new protein has to be synthesized. The other feature of this has toxicokinetic issues because of its irreversible nature. If one is being continually exposed to electrophiles, depending on the synthesis rate of the target protein, the modification can accumulate over time, so that even though the concentration may be low, depending on the target protein, its effect could be cumulative, and over a protracted period of time, the effect could be physiologically or cellularly significant. And an example of that is this regulatory protein KEAP1, which is alkylated by electrophiles, resulting in an activation of the antioxidant response element and the expression of heme oxygenase. And this is a common reaction with some of the nucleophiles that we have found, or sorry, electrophiles that we have found in ambient air. Alternatively, however, the electrophiles can react with I-kappa B in the same way that the reaction has KEAP1, but in this case, induce an inflammatory response. So depending on the target proteins that are affected by reactive oxygen species or electrophiles, one can see either an anti-inflammatory or an adaptive response or an inflammatory response, and that depends on the nature of the stimulating so-called reactive species and its concentration. The way we assay for these reactive species then is the pro-oxidants are assayed by what we call a DTT assay, and we perform this assay in the presence of a chelator, DTPA, in the presence and absence to separate then a metal-based pro-oxidant activity from a organic pro-oxidant-based assay. The electrophiles are assayed by our GAP-TH assay, which I'll explain in a minute. And then for, as I said earlier, for the anti-inflammatory or adaptive response, we've used HO1 as a marker, and for the inflammatory response, TNF-alpha. So the DTT assay then is an assay that essentially determines the consumption of dithiothreatol by an oxygen-dependent process, and it occurs both with organic pro-oxidants and with metal pro-oxidants, such as the redox active metals. But the electron transfer reaction with the redox active metals can be blocked with the metal chelator, diethylene triamine pentacetic acid, or DTPA. So by doing the assay in the presence and absence of DTPA, we can separate the metals from the organics. The electrophiles then are measured by measuring the changes in the activity of the enzyme glyceraldehyde 3-phosphate dehydrogenase, which is the thiolate enzyme, and its alkylation results in its inactivation. So we simply assay the residual activity and determine how much is lost, and the quantity of electrophile, of course, is proportional to its ability to inactivate the enzyme. And we perform these assays on an aqueous suspension of the particles, not an extract, but it's the particles themselves, and the results are then normalized to the volume of air. This is a slide that shows sort of the range that of DT activities that we have found in assaying samples from different locales, such as Beijing, Mexico City, someplace at a site next to the 110 freeway, as well as one in San Bernardino, the nature of which I'll be explaining in a minute, and Irvine itself. And as you can see, the Irvine DTT activity was very low, but if one concentrates it by a factor of 12, which is what one of the early basis concentrations demonstrated, then we would increase the activity to that which is comparable to Beijing and Mexico City. In the presence of DTPA, then much of the activity drops, and most of the activity, for example, in San Bernardino, all of it was due to metals, because the activity is essentially stopped or inhibited completely with DTPA. In contrast, Irvine, of course, had significant amounts of non-metal DTPA, DTT activity, sorry. I show this slide to show you the 3 locations from which we collected samples in a previous study at the rail yards in 3 communities: San Bernardino in the east end of the LA basin, Commerce right in the middle of downtown, and in Long Beach along the harbor. And we collected samples close to the rail yards at these 3 different locales. And we also collected samples in Commerce, but neighboring the Commerce rail yards. And just for reference, then this is where Irvine ends up on the map of the basin. And this slide just summarizes then the DTT activity for the samples in the absence and presence of DTPA. Now, one of the really outstanding changes or differences you note is, of course, the fact that all of the other sites, the non-Irvine sites, had virtually no organic pro-oxidants, whereas the Irvine samples all demonstrated significant levels of organic pro-oxidant content. Now, the significance of this, at least from our perspective, based on our previous experience with these systems, is that the organic pro-oxidants tend to be more pro-adaptive rather than pro-inflammatory. And studies that we've done with the rail yard samples would indicate that it's the higher metal, high DTT activity samples, most notably the Commerce and San Bernardino samples, as well as the one site of Commerce that's right next to 2 rail yards. They were the most active, and they were the only ones that were capable of eliciting a pro-inflammatory response. All of the other responses tended to be either minimal or pro-adaptive. And so, based on this, we did not do the cell studies on these samples. We have problems with our cell culture facilities, but the indications are, based on this, the indications would be that we would not expect to see a very strong pro-inflammatory response from the Irvine samples. So to summarize the results in terms of what we found and what Bill has described, the hypothesis that we were testing was that the exposure to concentrated ultrafine particulates would affect the progression of the Parkinson's-like, disease-like, and neurochemical changes in the transgenic animals. The results that, based on our measurements, indicate that our concentration was appropriate. We increased the concentration of the particles by a factor of 12 to something approximating that which was found for Mexico City. And the reason I indicate Mexico City is because a report from Mexico City indicated that the particles from Mexico City were involved in some of the early brain pathology that was observed in children and young adults by investigators in Mexico. We did not see changes in the significant changes in the inflammatory and neurochemical markers, as well as alpha-Syn. And we did see some changes in this one behavioral response. And the other feature that we found was the fact that the PM 2.5, or the ambient air from Irvine, was qualitatively different from that from other LA basin sites because of its high level of organic peroxidants. and lower levels of metal peroxidant content. And again, I say that because we, on the basis of prior studies, we think that the organic peroxidants tend to be pro-adaptive. So we may have expected a reduced level of the toxic response. So to put the observation in another way, the data would suggest that metal content may be very important in eliciting inflammatory responses, and we need to investigate that further, I believe. And the high proportion of organic components in the Irvine samples may have attenuated the inflammatory response that could have contributed to a greater or a more significant difference in the animal responses. So again, we should measure we feel that metals are important, and we need to examine them for their effects, and we're trying to set up things where we can do some of these kinds of studies. I'd like to acknowledge the help of all of the people in the different laboratories, from the climate laboratory, Andy Kibo, David Herman, Amanda Ting, Samantha Rush, and Laura Polowski, whose name I didn't include, and then from the Burton Jones Laboratory at Irvine, and this is the laboratory that provided us with the transgenic animals, and Natalie Goldberg was a graduate student who told us what to do with respect to the way these animals are to be treated. At UCLA, Deborah Schmitz did all of the analyses, and Lori Miyasato at CARB was our contract manager and advisor, and helped us immensely in the course of this project. Thanks for your attention, and I think I'll turn it over to Lori, who may have some questions for us.
Great, thank you very much, Art, and Mike and Bill and your entire team. It was a very nice and well-coordinated presentation. So it looks like we might have a question in the room. So, and for anyone who has questions, if you could please state your name and wait for the mic to come around to ask your questions, because this is webcast. Okay.
Stephen Renkus from Department of Pesticide Regulation. I was unclear about the time between the end of the exposure to the ultrafine particles and the sacrifice of the animals such that you could do a brain evaluation. How much time had elapsed?
Following the 22 weeks of exposure, the 23rd week was dedicated to behavioral tests, and 7 days, and then the next week, which would be 7 days after their last particulate exposure, the animals were sacrificed. So 1 week after the 22 weeks.
Yeah. So let me ask, is it possible that in that 1 week, while you were, of course, diverting, they were delayed so that you could do this live testing while they're alive, that things dissipated in the brain?
Yeah, it's possible. Usually the criticism is the other way around, that they think we were sacrificing, we're maybe artificially seeing an effect that didn't persist. We felt that after 22 weeks, those deficits, if any, should have been persistent. So the exposure conditions didn't vary over that time. So in theory, you could do it. You would like to do a time course, 1 day, 7 days, 2 weeks out. But we felt that 1 week would capture the cumulative effect of the exposure, and we were interested in a persistent effect. So in terms of working backwards, the alpha-synuclein accumulation would not have gone back down had it been induced and increased due to the exposure. As to the other markers, from our prior experience, following an insult, those at least take a couple weeks to recover, say in terms of dopamine or norepinephrine content decreases. We have done drug studies with methamphetamine in the past. So, so we felt that the 1 week time point would have more persistent changes in brain due to the.
One last question is, did you do any histo on the brain? And for example, the whole, the idea that possibly, besides going through the lungs, this stuff could pass through the olfactory, an intranasal passage into the brain. I think that's been shown for PM 2.5 particles and also from manganese oxide particles generated from welding. Do you have any sense if have you done anything along that lines?
Well, we considered that, but given the number of assays that we needed to do and the brain material required to do them, we just couldn't subdivide it any further. So we couldn't even hemisect. So that would have required hemisecting the brains and using 1/2 for histo and 1/2 for all our analyses. But we had 5, postmortem tests that require, a fair amount of material. So it just wasn't plausible to combine in this study. So we opted for the measures that we thought were more amenable to group analysis, because with 90 animals, it would be it would be a challenge to do histo on those.
Sure. All right, thank you.
Hi, and I'd just like to remind our viewers online that you can email questions to my address, lori.miysato@arb.ca.gov. Do we have any other questions? Okay, just a moment.
Thank you. This is Linda Smith-Carb. I wanted a few more details on the exposure itself. Since you had 4 cohorts throughout the whole year, I'm assuming that there was no seasonality, summer, winter, fall, spring. It was just throughout the year?
Bill, if you go back to that slide where you had the cohorts,
Mike.
All right, so this shows the time frame. So if you see, as you see in cohort 1, exposure week 1, we started in July, and the last exposure was the following January 18th.
Okay.
So those dates, but like I said, the exposure were constant for all 4 cohorts for a couple weeks before, at the beginning and end. And we just had no other solution than this. This we thought was challenging enough in itself, just to get the age-matched animals in sufficient numbers to generate sequential cohorts with reasonable time to allocate for the sacrifices. So this was sort of our best compromise on that. So essentially, they got a summer exposure, a late Southern California time period exposure.
Okay, thank you. That's fine. I also had another question regarding the air itself. I mean, clearly it's different from being near rail yards or Beijing or Mexico. But can you give any more details on the air? Was it more rural or agricultural or suburban, or what can you say about the air itself?
Mike, do you want to comment on that? Mike?
yeah. So this is, the site was at the University of California, Irvine. We are about a 1/2 mile from the 405 freeway and about, 6 miles from the coast. For the most part, we have a non-industrial suburban area with a lot of commercial operations, but not too much industrial. I would say it's it's more of a commercial area with a large amount of traffic, but very little heavy industry sort of input. However, the way the air moves, we have, when we do trace metal analyses, we can see evidence of emissions from the Port of LA, high amounts of vanadium and nickel on certain days. So depending on what's going on, we have a variety, a fairly mixed regional exposure. But it's not, there's some agriculture, much less than there used to be. And mostly, I would say the major input is from traffic.
Oh, thank you. And again, great seminar. Thank you very much.
Thank you. Are there any other questions in our audience here? Also, if there are any questions from Irvine or UCLA, feel free to chip in as well.
Well, actually, Bill, I had one question. Just looking at the data, it hadn't struck me before that the alpha-synuclein mice had higher levels of heme oxygenase, wild type. And I was wondering if that had been seen in other studies.
I don't think anyone else is looking for one. So we're going on with this readout.
It didn't seem to matter much with the behavioral measurements, but I was wondering if they were afforded a little bit extra protection against radicals by having a higher level of heme oxygenase, showing that their antioxidant response.
No, I mean, yeah, I guess I haven't really followed heme oxygenase or its changes in the brain very much. So I can't comment on that either, Mike. Are there any other questions?
And Art, we don't seem to have any from online. Are there any other questions from the room? In that case, we'd like to thank you all for a very nice presentation. Thank you.
Thank you.